Hello, everyone, and thank you for stopping by before the end of the day. My name is David Bazaga, and I'm the product lead at Kibim. For those of you who are not aware, Kibim is a Spanish-based company headquartered in Valencia, and we developed AI algorithms for the clinical practice. And the focus of today's call is going to be on QP prostate. So if you're interested in prostate MRI, hopefully you'll learn something interesting today. So just a bit of background around what we actually do in Kibim and what our business model looks like. We really try to transform imaging data into actionable predictions. And we do this in the full spectrum, going from the population level all the way to the patient level. What this really means in practice is that at the beginning of this cycle, we partner with pharmaceutical companies to really try to make the best use of their imaging data. So this can be looking at things like best treatment selection for a specific patient population, looking at prediction of patient outcomes with imaging data, or patient stratification. What we usually do here are research-based activities that then help biopharmaceutical companies further guide their clinical design processes and their clinical studies. And you can see here two publications as an example, both in cell lymphoma as well as biochemical recurrence for prostate cancer. Now, when these scientific ideas, we actually see a potential and we see a market fit, we take them through our life cycle process until they actually become products that can be marketed and can be commercialized as part of a hospital healthcare ecosystem. And the one that we will be focusing on today, it's QP prostate, as I mentioned. So just to provide a bit of context, in case you're not aware on how prostate cancer looks like, the journey for a prostate cancer diagnosis usually actually starts with the patient reporting symptoms himself. There's no national screening program like in breast, for example. So you usually start with the patient reporting symptoms to their general practitioner, and they would order something, a PSA test, which is a blood test. If that PSA level is high, they would go into an MRI that gets assessed by a radiologist. And if there are any suspicious findings there, then they would go for a biopsy. Now, there are two main bottlenecks in this pathway. The first one is around this PSA test that I mentioned. This test has suboptimal sensitivity and specificity, meaning that there are people with cancer that show us negative here, but there's also a lot of unnecessary onward referrals for MRI. There's plenty of evidence that suggests that the PSA screening program is not fit for purpose. The highest one is the one from the Reimagine group in the UK, led by UCL, where they actually showed that upon MRI, half of the patients with prostate cancer actually had a negative PSA. So they would have gone unnoticed had not been from the MRI. The other one that I've mentioned there is from a Canadian trial showing very similar results. And the second bottleneck is in the actual MRI radiologist pathway. So prostate MRI, it's usually characterized by long reporting times, high inter-reader variability, and obviously a radiology shortage widely reported in all other disciplines, not just prostate. I would like to delve in the latter two. So in terms of inter-reader variability, just to really exemplify how the disparity across scores, what you can see here on the y-axis are 26 different hospitals from the US. And what you see in the x-axis is the positive predicted value. So that is when the radiologist is saying that they believe there is prostate cancer, how likely that prostate cancer really is to be. And you can see that really depending on the hospital that you end up, that probability goes from 20% all the way to up to 70%. This obviously creates a postcode lottery that really depending on the hospital that you go, you actually may undergo an unnecessary biopsy, miss cancer, or if you actually get lucky, the proper diagnosis and the further diagnostic treatment that you would like to have should you have prostate cancer. The second one is around radiologist shortage, and I think everyone here in the room is familiar with the ongoing and growing radiology shortage that we've got in Europe, in the US, and worldwide. Here is the projection from 2020 around the gap between the availability of radiologists versus the availability of them, and we can see that that gap is only going to be growing over time. Just to exemplify what this means for prostate cancer patients, this is some data from NHS England in the UK, and you can see that only roughly half of the prostate cancer patients get diagnosed within 28 days, which is the national target. Now, that target should be around 75 or above, but for prostate cancer, that currently sits at 55%. And obviously, this is due to a lack of radiologists, but also a lack of inefficiencies throughout the whole prostate pathway that I just mentioned. So with this landscape and with these bottlenecks, what we've developed at KIMM, it's QP Prostate, which is a solution designed to help radiologists in the reporting of prostate MRI. And what we do here, first of all, is a series of pre-processing steps to really make sure that all the data is fully aligned in terms of diffusion, tissue, and contrast imaging sequences. The second thing we do is a quality check against PIRATES guidelines. For those of you not familiar with PIRATES guidelines, this is a set of international guidelines setting how images should be actually acquired. What we do here is really check if those studies are really PIRATES compliant or not. That does not necessarily mean that we don't work with non-PIRATES compliant cases, but this is something that we provide back to the radiologists. We also provide a prostate segmentation, distinguishing between the peripheral and the transitional zone, as you can see there. And last, but certainly not least, we provide a fully automated lesion detection for the clinically significant prostate cancer. You can see an example on how this looks as a secondary capture that gets sent back to the PACS. So you can see the prostate segmentation on the left and the lesion detection on the right. So this information gets fully automated, sent back to the PACS so that the reporting radiologist can visualize all this information at the time of reporting and incorporate it into their clinical thinking process. In terms of how we actually developed and tested this tool, what we do here is take a biparametric input, so that is the T2 weighted and the DWI sequences. We generate our own ADC and high-B value maps, and the lesion detection algorithm is essentially made up of 3 different steps. The first one is a clustering algorithm, which essentially takes different or similar regions within the prostate based on the intensity of the T2 and the DWI images. That gives us a list of potential candidate lesions that then we need to identify whether they are prostate cancer or not. That is the second step in classification, and for that we use biopsy as the ground truth to really determine whether a particular lesion was prostate cancer or not. And finally, we provide a post-processing algorithm that provides a risk score depending on the certainty the algorithm has on whether something is prostate cancer or not. So what you can see here are 2 examples of what I've just mentioned. We provide a 2-tier risk system. The orange one that you can see above, it's what we call a high risk or a high certainty lesion. So these are essentially lesions that the software believes are high likelihood to be prostate cancer. And below you can see a lesion highlighted in yellow, what we call moderate risk. And what we really tried to do here was try to mimic the clinical thinking process when assigning a PIRATES score, which is essentially a level of confidence on whether something or that particular lesion could or could not be clinically significant prostate cancer. It is also worth noting that we also distinguish where that lesion is located within the prostate. The prostate tumors look very differently in the peripheral zone versus the transitional zone. So this is something that we have also incorporated into our algorithm to really make sure that we get the most accurate results. And now I would actually like to present some performance data from a preliminary study that we've performed with Mass General Hospital and their data science office, really to look at the standalone performance of QP Prostate. This was a retrospective cross-sectional evaluation looking at both 3T and 1.5 and different vendors. So we try to do it as comprehensive as possible with the data set used for this evaluation. We selected the cases with a 50% enrichment, so that means 50% were positive and 50% were negative. The negative cases could actually come from biopsy confirmed or non-biopsy, but we followed them up over a year to really ensure that they were truly negative cases. There was a quality assurance check to really ensure that the cases were PIRATES compliant, that there were no artifacts, or that they were actually of good diagnostic capability. And then we ran them through the model, as well as through a ground truth panel of 3 different radiologists, where they could actually segment the lesions, and then everything was compared against biopsy data. Before jumping into the results, I would like to take a moment to actually explain what the metrics presented are. We present them both at the case level or patient level, as well as the lesion level. So at the case level, we're really looking at a triage solution where the software may or may not provide a positive output, and the patient may or may not have prostate cancer. But here we're not specifically looking at the localization of the software with the right lesion. So this may end up in a situation like the one you can see at the bottom left of the screen, where the real lesion and the software prediction are in completely different places, but we would take that as a true positive from a triage tool perspective. We also present lesion level results, and for these ones, the localization of the software prediction obviously had to coincide with the ground truth annotation and the positive biopsy results. And these are the results. They're obviously presented for both the high marker, so that high certainty that I mentioned, as well as the moderate marker, so the less certain ones. And you can see the model achieved in a completely unseen database an AUC of 0.747. The sensitivity obviously increases with the moderate marker. We go from 90 to 95%, but obviously that goes at the expense of the specificity. So the specificity for the high marker, for that sort of equivalent to a PIRATS 4, PIRATS 5 lesions, stands at 57%. When we go to the moderate, so sort of like a PIRATS 3, that drops to 38%, which this is really in line with what expert radiologists or common radiologists usually report when compared to a biopsy. When we go to the lesion level results, the sensitivity, as you can probably imagine, drops down, because obviously now we really need to properly locate the lesion in the right place as per the ground truth segmentation. So the sensitivity here dropped to 77% for the high marker, and for the moderate marker, it actually obviously increased to the 88%. Again, similar to that, the drawback here, it's in the false positive rate per case. So for the high marker, we essentially output 0.4 false positive cases per prostate. And for the moderate marker, that goes up to 1 false positive rate per prostate. Now, you may be thinking that this may look not too great in the sense that I'm giving 1 false positive for every single prostate that I'm looking at, but that doesn't necessarily mean that they're all happening in the same prostate, which is why the results at the case level are also relevant here. What you can see here as well is the free response ROC. So this is essentially a ROC curve, but also taking the localization endpoint into account. And the equivalent of the AUC metric at a lesion level comes at 0.7, which is very in line with what the case level results were also indicating. I also wanted to highlight here what this really means in terms of clinical practice. And this is really how much can a radiologist trust the output that we are providing here. So I would like to draw the attention to the fact that obviously PPV and NPV values are dependent on the cancer prevalence, but for most prostate cancer pathways, you're looking at a prevalence between 10 to 20%, which is highlighted by the vertical lines that you can see there. And what you can really see there is that the negative predicted value, so this is essentially saying when the algorithm is telling you I haven't seen anything, how much can I trust it? Even for a prevalence of 20%, which is relatively high, that NPV is around 97, 98% for both the high and the moderate marker. And what we essentially want to highlight and convey to the radiologists with this is that when the software has not found any lesions, you can really trust the fact that there is no prostate cancer here, and thus avoid any unnecessary biopsies that you may potentially avoid further down the pipeline. And obviously, this is just a pilot study, a clinical evidence generation. It's an ongoing process. So in terms of next steps, we want to perform, or we're already performing, a larger study in collaboration with Mass General with a larger sample size. We're also performing a multi-reader, multi-case study. So that is essentially looking at not just how good is the AI on its own, but how good is the AI helping radiologists. So what we'll be comparing here is radiologists plus AI versus radiologists on their own. And the results of that will also be ready within the next 4 weeks or so. And obviously, we also want to evaluate this in other populations. There is a big push for AI tools in MRI screening, similar to what is happening in breast. That is obviously a very, very different population from a prevalence perspective, but also from a disease etiology. So that is something that we will also be researching in KBIM as a further future offering. So that's everything that I wanted to go through. Thank you very much. And if you have any questions, please feel free to ask me now or stop by our booth, which is literally as you leave here to the left. Thank you.
Thank you. Thank you very much. Are there any questions? I might have a question then. Very much looking forward to the results of your multi-case, multi-reader study. That's in the end going to show the impact in a clinical setting. As most of the times, as you showed, there's always a balance between sensitivity and specificity. Your tool is clearly sensitive, less specific now, as a standalone.
Yeah.
How do you rate that trade-off of being more sensitive versus and less specific in terms of clinical impact and radiological workflow?
Yeah, no, that's a great question. And I think the key thing really is depending on the radiologist's profile. So what we usually try to advise here is that for most inexperienced radiologists who are just getting started with prostate, they should really take a closer look to those yellow lesions that we highlight, because that's really ensuring that you're not missing any cancers, even though that's at the expense of unnecessary biopsies, but that's part of that learning curve. When you're looking for more experienced profiles of people doing prostate MRI, they may just wish to look at the orange ones, which are the high certainty ones, just really to ensure that nothing is being missed. But from a false positive red perspective, they're obviously able to evaluate what the AI algorithm is telling them and being able to discard it should they wish to do so. So it's obviously depending on the level of experience and also the use case that you would like to see. Some radiologists actually tell us that they don't mind having a lot of false positives because it's their job then to downgrade them and say, I disagree with it, but I appreciate that the software is flagging them to my attention. So from that perspective, low specificity is actually preferred from a patient safety perspective. So it really does depend on the profile of the radiologist, the clinical practice on that given hospital, and what they're really looking for. And that's why we provide both of them, depending on what their needs are.